Good morning, good afternoon, and good evening, everyone. And welcome to the Scientist.com webinar titled Exploring Predictive Biomarkers and ERK1-2 Phosphorylation, a New Horizon in Forecasting Survival and Response to Immune Checkpoint Blockade in Glioblastoma Treatment. I'm Liam Sanyo from the events team here at Scientist.com, and I'll be your host today. Big thanks to Akoya Biosciences for helping to sponsor this event. Wouldn't be possible without them. We're very fortunate to be joined by Dr. Victor Arrieta, postdoctoral fellow in the Department of Neurosurgery at the Northwestern University Feinberg School of Medicine. Today, Victor will present his research highlighting the link between p-ERK activation and improved survival in recurrent glioblastoma treatments using anti-PD-1 immunotherapy. And without any further ado, I'd like to welcome Dr. Victor Arrieta. Victor, thanks so much for joining us today, and the floor is yours whenever you're ready.
Perfect. Thank you very much, Liam. Okay. Thank you for this opportunity to talk about the clinical research that we have done in our group. Yes, I am Victor Andres Arrieta, and I am a postdoctoral fellow working at the laboratory of Dr. Adam Sonovan at Northwestern University. And today, I'm going to talk about biomarker that could be potentially employed to identify patients with glioblastoma who are likely to benefit from immune checkpoint blockade. So to start, I would like to mention that immune checkpoint inhibitors have seen an unparalleled expansion in clinical oncology in the past decade. Given the remarkable success and enduring clinical responses seen in many of these cancers, immune checkpoint inhibitors targeting the PD-1 receptor suggested an encouraging opportunity for glioblastoma. Importantly, I would like to discuss the results of recent phase 3 clinical trials that evaluated the effects of PD-1 blockade in both newly diagnosed and recurrent glioblastoma patients. In this study that was published recently, there were no survival differences between the nivolumab group, as you can see here, and the bevacizumab group. However, despite these negative findings, various other studies and case reports have indicated that certain groups of patients do show lasting radiographic responses and extended overall survival after receiving this anti-PD-1 immunotherapy. To illustrate this phenomenon, I would like to draw your attention to these two cases, both of which were treated with adjuvant PD-1 blockade. The first non-responder patient, as you can see here, experienced recurrence of the disease, then had tumoral resection. He went to the OR, and then started anti-PD-1 immunotherapy. You can see here that the tumor recurred, and the patient passed away after 6 months of treatment, as you can see here. In contrast, this other patient had this nasty-looking tumor that was resected and then started PD-1 blockade. What was interesting is that during active treatment with immunotherapy, this patient developed an enhancing lesion, as you can see in this MRI, and a biopsy was obtained from this lesion that revealed abundant T-cell infiltration, as you can see here. And this was in the lesion, and it developed a similar lymphocytic pattern in the CSF. And then this patient continued with the immunotherapy for a total of 21 months from recurrence prior to being lost to follow-up. So what is different between these patients? One clinical study characterized some of the genomic hallmarks associated with response to PD-1 inhibitors in recurrent glioblastoma. In this study, GBM patients treated with either pembrolizumab or nivolumab were classified as responder patients or non-responders, and they had to meet at least one of the criteria to be classified as a responder patient. So the first one was that tumor volumes as seen from magnetic resonance imaging were either stable or shrinking continually over at least 6 months, and second, that the tissue obtained during surgery after PD-1 inhibitor grossly showed only an inflammatory response and very few to no tumor cells, and this is associated with pseudoprogression. After whole genome sequencing of pretreatment tumor samples, it was found that activating mutations in BRAF and PTPN11 were enriched in responder patients. Now, only 30% of the responder patients harbor these mutations, leaving the rest of the responder patients with an elusive molecular profile. Interestingly, as you can see here, these genes are members of the MAP kinase pathway, which implicates this cascade in response to PD-1 blockade. As you can see, all the MAP kinase signaling pathway converges in the phosphorylation of ERK1 and 2. Thus, we focus on p-ERK or phosphorylation of ERK as an indicator of the activation of this pathway. So first, we wanted to know and determine whether phospho-ERK was a prognosis or a predictive biomarker. To determine MAP kinase activity in each specimen, we perform quantitative image analysis of pretreatment tumor samples within tumoral regions that were selected by a neuropathologist who was blinded to the clinical data. And with this, we quantified the cell density of p-ERK positive cells. For this analysis, we had a control cohort that didn't receive immunotherapy, as you can see here. And also the cut point value used to classify high and low p-ERK tumors was derived from the median of all values for phospho-ERK cell density. So based on the initiation of PD-1 blockade at recurrence, GBM patients treated with immunotherapy demonstrated a longer overall survival compared to patients whose tumors were classified as a low p-ERK. You can see like the red curve is the ones that survive longer. In contrast, phospho-ERK was not associated with survival in patients who didn't receive immunotherapy, as you can see here in these curves. We also performed univariable and multivariable analysis using the Cox proportional hazards model to evaluate the association of phospho-ERK cell density with survival. This analysis revealed that phospho-ERK cell density was associated with increased overall survival in patients treated with PD-1 blockade in both univariable and multivariable analysis, but this was not demonstrated in patients that didn't receive immunotherapy, as you can see in these forest plots. We then investigated whether elevated phospho-ERK cell density and its association with long-term survival following PD-1 blockade. We wanted to know if this can occur irrespective of BRAF and PTPN11 activating mutations. Interestingly, patients that survive more than 12 months from initiation to PD-1 blockade had elevated phospho-ERK cell density compared to those that survive less than 12 months, as you can see in this comparison. These results show that patients who experience a longer overall survival in the context of immunotherapy consistently exhibited high levels of p-ERK regardless of the genetic status of BRAF and PTPN11 genes. So then we contacted our friends at UCLA, Dr. Robert Prince and Tim Clausi, as they were conducting a phase 2 clinical trial evaluating neoadjuvant PD-1 blockade. So in this trial, or in this cohort, we focused on the tumor samples from the arm that was treated with adjuvant immunotherapy and analyzed them with the same methodology and cutoff value used for the discovery cohort. So in this independent cohort, we found that patients with high p-ERK tumors had an increased survival compared to patients with low p-ERK tumors. These results were confirmed with a Cox proportional hazards ratio in which p-ERK was associated with survival as well. Next, we wanted to determine the immunological differences between these tumors. And for this purpose, we employed multiplex immunofluorescence using Bactra3 from Akoya Bioscience. We employed validated cellular markers in glioblastoma, including SOX2. We also use TMEN119 for microglia and CD163 as a pine myeloid cellular marker. So here we have two high p-ERK tumors, one of which harbors a BRAF mutation and one that is wild type for BRAF. In addition, we have also a low phosphorus tumor. So we quantified the main cell populations, as you can see here, and we found that SOX2, or our tumor cells, or that phosphorus was detected predominantly expressed by tumor cells relative to other cell types. In addition, an analysis focused on the myeloid cell compartment relative to phosphorus revealed that there was a high content of TMEN119 positive microglia in high p-ERK tumors compared to low p-ERK tumors, as you can see here. Also, there was a positive correlation between p-ERK and IBA1 positive cells that identifies both microglia and macrophages. And here we can see some representative images of microglia in regions that are high for p-ERK. Next, we performed spatial analysis to further investigate the interaction between SOX2 p-ERK positive cells and glioma-infiltrating myeloid cells. We calculated the distances between TMEN119 positive cells and CD163 positive cells to tumor cells expressing p-ERK and also tumor cells that were negative for p-ERK. And then what we found here is that TMEN119 positive cells were closer to SOX2 positive p-ERK positive cells in tumors that were classified as high p-ERK compared to the distance, which was a longer distance between these two cell populations in tumor samples that were classified as low p-ERK. Conversely, we didn't find differences between TMEN119 positive cells and SOX2 positive p-ERK negative cells, as you can see in this analysis. Here we have some representative images of shorter distances between these cell populations and larger distances between these cell populations in both groups. Perfect. So in a different panel of markers using GFAP to label tumor and astrocytes using the PhenoImager HT, formerly referred to as a Vectra Polaris, we measured the distance between tumor cells and myeloid cells. So we found that the distance from CD163 positive cells to GFAP positive p-ERK positive cells was shorter in tumors that were classified as high p-ERK compared to tumors that were classified as low p-ERK. We didn't find differences between these two cell populations when we evaluated GFAP positive p-ERK negative cells. We can see here some images to represent this analysis, where we have larger distances here between myeloid cells and tumor cells expressing phosphoric. So next, we analyzed the tumors from an independent cohort of 10 GBM patients using PERK single-cell RNA-seq and immunohistochemistry. We employ unsupervised clustering to create these human plots, and then we classify tumors as either high p-ERK or low p-ERK using the same methodology as the discovery and the validation cohort. We annotated the cell clusters based on the expression of validated cell markers for glioblastoma and single-cell transcriptomics. So here we can identify tumor cells, we can identify myeloid cells, parasites, and endothelial cells. And then we label all the cells based on whether they were coming from either high or a low p-ERK tumor determined by immunohistochemistry. So here we focus on the myeloid cell compartment, here, as you can see here, based on the abundance seen by multiplex immunofluorescence. And we ask ourselves, what is different between myeloid cells coming from high and low p-ERK tumors? To answer this, we perform differential gene expression analysis, comparing the myeloid cells populations. And among several of these upregulated gene signatures, we found that MHC class II protein complex binding was the topmost enriched in myeloid cells derived from high p-ERK tumors. And here we can see like a representative UMAP plot and another plot that shows this enrichment in myeloid cells derived from high p-ERK tumors. So then, to validate this finding, we use multiplex immunofluorescence staining against a component of the MHC class II molecule and found that this was also detected at the protein level with TMN119 in magenta here and MHC class II in white. This suggests that myeloid cells from high p-ERK tumors have better antigen-presenting capabilities, or that these cells are in a state of activation that may promote a better response to PD-1 blockade. Next, we sought to identify and further validate this biomarker in a phase 1 clinical trial done in Belgium, in which recurrent GBM patients were treated with 1 dose of nivo preoperatively, so you can see here, followed by intracerebral administration of nivolumab and ipilimumab, and followed also by adjuvant nivolumab twice a week after surgery. Here we have the demographics of this cohort. I want to note here that we have 25 IDH wild-type GBM patients and 2 IDH mutant patients. And here we can see the survival curve in this cohort of this phase 1 clinical trial. So here you can see from the 27 patients that were enrolled in this clinical trial, we analyzed 24 as 3 of them were not acceptable for immunohistochemistry. Similar to the previous cohorts, we used the median value to dichotomize this cohort into high and low p-ERK groups. And as an exploratory analysis, we also divide the cohorts into 3 groups. Lastly, we excluded the 2 IDH mutant patients and performed a focus analysis on IDH wild-type GBM patients. To increase the rigor of this analysis, we followed the remark criteria in which we pre-specify the conditions for analysis a priori. Then we analyzed, then one of the teams sent the clinical data, in this case, the team from Brussels. They sent the clinical data to the biostatistics team, and another team quantified the biomarker levels in a blinded manner. And finally, the biostatistics team performed the survival analysis. Here, when we analyze all the patients, including both IDH wild-type and IDH mutant patients, we found a trend towards statistical significance favoring survival in the high p-ERK group. However, when we focus our analysis on IDH wild-type GBM patients, we found that those patients with high p-ERK tumors had a statistically significant longer survival than those with low p-ERK tumors. And with that, I would like to leave you with these conclusions. So ERK1 and 2 phosphorylation shows that it is a potential predictive biomarker of immune checkpoint blockades, including anti-PD-1 therapy, now in 3 independent cohorts of recurrent GBM patients, that these tumors, the tumors that exhibit high numbers of p-ERK positive cells, contain abundant microglia, and myeloid cells derived from high p-ERK GBMs exhibit increased MHC class II expression compared to myeloid cells derived from low p-ERK GBMs. Although we found an association between p-ERK and survival in the context of immunotherapy, we remain cautious about these findings. And in fact, we believe that some aspects that need to be considered and taken into account before implementing this biomarker for anti-PD-1 therapy. So we believe that there should be further validation in additional cohorts because we had a limited number of samples. We choose a cut point value based on the median, but we believe that with higher numbers of patients, cut point value optimization should be required. Also, alternative techniques for activation of the MAP kinase pathway will be useful, and standardized protocols for p-ERK assessment will be also important. Lastly, I want to thank all the co-authors of this work, and special thanks to my mentor, Dr. Adam Sonovan, and also special thanks to Akoya Bioscience and the team that made this possible. And thank you very much, and happy to ask and to answer any questions.
Excellent. Well, thank you, Victor, for a really great presentation. We'll move right into a Q&A. And as a reminder, if you do have a question that you'd like to ask, you could submit it using the Q&A tab on the right-hand side of your screen. All right, let's jump right in here. So, Victor, what might account for the varied responses to PD-1 blockade in glioblastoma patients, given that some show no difference while others experience prolonged survival?
Okay, thank you, Liam, for all this introduction and everything. And well, that's a really good question, and one that underscores the complexity of the tumor microenvironment and patient heterogeneity in gliomas. Now, something that I want to mention before answering this question is that based on the previous clinical trials, as well as studies that have been performed in our group, we believe that responsive GBM patients do not represent a big proportion of all patients. However, we believe that the accurate identification of this small subset of patients could provide them with a meaningful clinical benefit. Now, several factors might contribute to the varied responses to PD-1 blockade. Particularly, we believe that cellular states and the interaction between tumor and immune cells might have an important contribution to immunotherapy. For instance, the presence of certain immune cells, such as T cells, or a specific subset of myeloid cells, like microglia, activated microglia specifically, can promote or inhibit the efficacy of PD-1 blockade. And another important factor, lastly, that contributes to these varied responses to PD-1 blockade is the overall immune health and profile of a patient. For instance, factors like previous treatments, immune cell count, and other concurrent illnesses can also modulate this efficacy. For instance, in fact, one of the hypotheses that we have is that p-ERK might be a reflection of the influence that the tumor immune microenvironment has on the tumor phenotype. And this is based on previous studies led by Toshiro Hara and Itai Tiraj that have been shown that myeloid cells can modulate the phenotype of glioma cells to become more immunogenic. So it is important also to consider these factors.
Excellent. Great answer. All right. Given the potential of p-ERK as a predictive biomarker, how do you see this influencing patient selection for anti-PD-1 therapy in the future? And what are the potential implications for improving outcomes for these recurrent GBM patients?
Well, I believe that the identification of p-ERK as a potential predictive biomarker is exciting. I think if its role is validated further, and it could support patient selection for anti-PD-1 therapy. So instead of one size fits all approach, we will be able to selectively treat glioblastoma patients most likely to benefit from this therapy based on their p-ERK levels. Right now, we are working on a project in which we are trying to use methylation data to determine the activation of the MAP kinase pathway. I think the advantage of using methylation data in addition to immunohistochemistry is that is now widely available in several institutions in the country. And in addition, it overcomes any issue with tissue processing that sometimes can be a challenge for immunohistochemistry. And finally, leveraging advancements in bioinformatics and in computational techniques. The rich data from methylation profiling can enhance the precision in identifying patients likely to benefit from immunotherapies.
Yeah, very cool. Exciting work. How do you see the role of multiplex immunofluorescence and single-cell RNA sequencing evolving in future glioblastoma research, especially in relation to deciphering tumor immune interactions?
Well, yeah, I think that will be very important for mostly for the effects derived from new therapies. I believe both multiplex immunofluorescence and single-cell RNA-seq are transformative tools for sure in glioblastoma research. I think multiplex immunofluorescence provides spatial context which is vital to understand the interplay between tumor cells and the immune system. In the future, I foresee a more integrated use of these techniques. I think combining the spatial resolution of multiplex immunofluorescence with the granularity of single-cell RNA-seq can provide a comprehensive picture of tumor-immune interactions. And something also to mention that is very important is that over the past few decades, numerous clinical trials targeting glioblastoma have aimed for just positive clinical outcomes, which is very important, of course. Yet many of these studies lack correlative analysis to ascertain effective drug concentrations in the brain or assess the experimental drugs' impact on the tumor and its immune microenvironment. So I believe that these technologies will be instrumental in evaluating the biological efficacy of upcoming therapies in glioblastoma patients.
Yeah, definitely. Excellent. Thanks a lot. Next question here. Are there any other potential biomarkers that could be used to predict response to immunotherapy in GBM?
There are some that have been described and published in the recent years. Unfortunately, several of the biomarkers that are used for other cancers have not been effective for glioblastoma patients. And these include, let's say, mutational burden, PDL1, um, even, and so some of these have not been very effective, but very recently, for instance, there have been some insights like indicators of the DNA replication stress that was published by Daniel McCrail recently, which suggests that this could be a potential biomarker. Also, there are some reports that shows that mutations in the pol gene can be particularly important for response to immunotherapy, where there are some glioblastoma patients that have exhibited incredible responses to these immunotherapies, but this still needs to be consolidated as well, I believe.
Definitely. All right, another great question here. Did you use a set multiplex immunofluorescence panel, or did you create your own? And what were the most critical factors you considered when developing this multiplex immunofluorescence panel for glioblastomas?
Well, we developed our own panel for multiplex immunofluorescence, and this was done in collaboration with the help of the immunotherapy assessment court with Surja and Megan and Rishita. So we developed it here as a team, and I think one of the or several critical factors for the developing of these markers were I would say the specificity and sensitivity of the antibodies. Those were very important. Also, the tumor heterogeneity was a very important factor to use multiplex immunofluorescence, especially for glioblastoma. We also considered, obviously, like the hypothesis that we had to then answer and evaluate the questions that we had to specifically for p-ERK and the assessment of the tumor immune microenvironment. And another important aspect of this technology is that Akoya has this technology that is able to amplify the signal for specific markers. And this was very important for us, particularly for microglia, to identify the abundance of microglia. So that was also super useful when developing these panels.
Awesome. Another one here. Can you comment about tumor heterogeneity and PD-1 blockade in glioblastoma? And also, can you comment about the value of multiplex immunofluorescence in studying this heterogeneity?
Definitely. Yes, as we have been discussing, these tumors are characterized by remarkable intratumoral heterogeneity and even intertumoral heterogeneity, both at the genetic and the cellular level. And we believe that this heterogeneity can influence therapeutic responses, not just for immunotherapy, but for other types of therapies. So for instance, taking into account these factors, we believe that some regions of the tumor might be more immune responsive than others, leading to varied outcomes with immunotherapy. And I believe that multiplex immunofluorescence allow us to visualize multiple targets simultaneously within the tumor microenvironment, and also to capture the interaction between tumor and immune cells and even other stromal components. So I believe that this was very useful. Initially, we were not aware that that we were going to find something specific for microglia, and I think that that also sheds light on the role that microglia could have on response to PD-1 blockade as well.
Yeah, some really good points. Was the multiple immunofluorescence done on whole slide or regions of interest? And did you use an intensity threshold for p-ERK positive versus negative?
Yeah. So what we did for this is that, as you can imagine, several of our tumor samples would have normal brain or peritumoral brain regions, and of course the tumor tissue as well in the same slide. So we worked together with Dr. Craig Korvinsky, who is the neuropathologist and also the director of the tumor bank here at Northwestern University. And he helped us to delineate all the tumor regions in the slide. So we focused specifically on these regions. So what we did was to perform a whole slide scan, and then we focused specifically on these tumor regions that were selected by Dr. Horvinsky. And then with regard to the intensity threshold for p-ERK, so we use p-ERK. So we knew before, because we did immunohistochemistry against p-ERK. So we knew which samples were high on p-ERK. So we used these samples as a reference to then quantify and determine the parameters for the microscopy during the analysis of all tumor samples. So basically, we use positive samples for p-ERK, and then we use those parameters to analyze all the tumor samples.
Fantastic. Yeah, that makes sense. All right, it looks like we just have one last question here. Victor, what analysis software did you use for these multiplex immunofluorescence studies?
Yeah, so we use several techniques, but the most important one was Inform, to do the processing of the images. So Inform allow us to do the phenotyping, and then first we did like cell segmentation, phenotyping, and further quantification of all the main cell types in glioblastomas. And after processing the data with Inform, I did several analyses, but these were performed in R, and these were so these analyses were done to mostly visualization of the cell types that we had in our tumor samples. But the primary software was Inform, and followed by different R analysis.
Perfect. All right. Well, Victor, thanks so much for joining us. It's really been a pleasure speaking with you today.
Thank you very much, Liam, for your time, and thank you everyone for attending.
Yeah, for sure. Big thanks to the audience here for joining us today. And in closing, we hope you enjoyed this scientist.com webinar, and we'll see you again next time. Have a great day, everyone.
Thanks.